any organs of attachment, right? So that's why it's important in treatment that you give something that will remove this attachment, right? Because if you see, because it can detach itself, right? Kalau you bagi ubat, it's still attached, so it can regenerate back. So it's important to give medication that can expel this colex also, right? But here, instead of suckers, right? Instead of the your suckers, the round suckers like in the trematodes, right? And with other things, here you have, it's not really a sucker, it's like a clip, right? This two leaf-like thing with a groove, so it's like it clips, right? So it's called a bothrium. So the proglotid can be as much as 3,000, right? So the mature segment is broader than long. So you see it's broad, because there are others which are longer than broad. But here, that's why the name also sometimes called the broad fish tapeworm. They labor and also long, right? But we're talking about each segment. Each segment though is broad, right? So each segment, mature segment, contains a bilobed ovaries. You can see the bilobed ovary, right? Just like, right? And then a coiled uterus, right? So all of those is something like a flower, right? It resembles a petal of flower or rosette pattern located in the center. So the reproductive organs like vas deferens and vagina situated in the middle, right? And here, which is a bit different from others, you don't see gravidity, you don't see ova, because they have a uterine pore. So near the uterus, there's a pore, there's a lubang there, which actually it pushes out the ova. In other tapeworms, they don't have this, so you'll see it's full of ova. But here, usually tak sempat like collect, right? The ova is being expelled, so you don't see much ova. It's just the reproductive organs there, the rosette pattern. So you can see a single worm can produce 1 million ova daily. Such high fecundity. So this is the term I want you to also remember. Fecundity, meaning a power of reproduction, right? So when we say, compare this, for example, to another worm, maybe produces just hundreds of ova per day, right? So it means the one with 1 million is more fecund, right? The fecundity is higher or greater, right? So look at the ova here, right? The ova actually resembles a trematode egg, if you remember. Even the size, 70 microns macam ni, right? It's like a perigonymous egg, or fasciola ova. And it is also operculated here, which you do not see in other tapeworms, in true tapeworm. So that's why Diphyllobothrium is somewhat like in between, right? It has eggs, ova, features similar with trematode, right? Its colex also leaf-like and so on. And also, see, remember when we were doing tutorial, we found the term coracidium, right?
Sama macam.
So here is somewhat like a trematode, right? After a while, in the right temperature and it will develop a coracidium. So something like a miracidium, right? The ciliated structure that swims, right? To find the snail host. But here, does it have a snail host? So here tak ada snail host. But, so a snail is a mollusk, right? Told you earlier. Part of a mollusk is hard, right? The snail. Part is soft, part is hard. But here, it utilizes the crustacean pula, right? Crustacean ni haiwan berkulit keras, macam ketam, udang. These are crustaceans. So we have a small crustacean as the first intermediate host, which is a cyclops. Many names to it: cyclops, water hyacinth, apa lagi? Brine shrimp. Even ada jual kat Toys R Us ni. There's brine shrimp egg, kan? Tu budak main-main. Jadi telur ni masuk air, it will hatch. You can see tiny sort of udang here, right? They make it into a toy. So this is the coracidium. So you can see the cilia is long. Usually the miracidium, the cilia is short, right? But still it's ciliated. So it has both features, right? Tapi tengok tengah-tengah ni, what is there? It's like something like right in the middle. Ada three things, right? It's actually three pairs of hooks. And so we call it hexacanth, meaning there are three pairs, tapi ada 6 hooks. Hexacanth embryo, right? So this is the hexacanth embryo. This is common to all Cestoda. Usually all the ova of the Cestoda will have this hexacanth embryo in the middle, like a rudimentary hooks. Tapi bila dia besar, there's no more hooks. Other tapeworms, they have hooks, and bila dia besar pun ada hooks. So it depends on the species. So it is something like having both features here, right? So the coracidium will penetrate the copepod or brine shrimp or water hyacinth or cyclops, right? And in there, it will develop into a juvenile stage, a larvae, right? In the trematode, it becomes the asexual stages, right? Rhedia, sporocyte. But here, it becomes a procercoid. So in the first intermediate host, right, within this crustacean, it develops into procercoid larvae, because there are 2 intermediate hosts now. So mostly in the trematoda, it's only 1 host, right? 1 intermediate host, except for a kinostoma, you have 2, right? And a few others also 2, right? But here It's totally different. You don't have snails here, so you start to have copepods. Within this, it is the coracidium. The procercoid larvae. And then these copepods are usually eaten by small fishes, right? Small fishes, right? Minnows and so on. Macam ikan bilis lah, tapi ikan bilis tu laut ni dalam air tawar, right? Minnows usually. So these small fishes pula, right? It will develop into plerocercoid larva, right? Procercoid within the copepod, and then within the small fish, it will develop plerocercoid larva, right? Within the fish meat, right? In the flesh, it will be the plerocercoid larva. And then here it is somewhat like a 3-stage, tapi just, right? And then the small fish will be eaten by a large fish, right? Salmon, trout, all these large freshwater fish in the temperate regions, right? Ini bukan tropical fishes, right? Rainbow trout, all this. So, but it doesn't change stages now. Tadi that procercoid becomes plerocercoid in the small fish. But when the small fish is eaten by the large fish, the plerocercoid doesn't change stages. So here you are introduced to a new term now, what we call as paratenic host. Meaning the host changes, right, from small fish to large fish, but there is no change of developmental stage. So this is what we call as paratenic host. So the large fish is the paratenic host, right? So this is the fish that is usually eaten by humans, right? Right? So you get infected if you eat raw or undercooked fish, right? Containing the plerocercoid larvae, right? So it is a zoonosis because it also completes its life cycle in the bear, for example, right? So the main thing for you as medical practitioners is the pathology, right? So the pathology mainly is megaloblastic anemia. So what is megaloblastic anemia or macrocytic anemia? The cells become large, the red blood cells, instead of small in microcytic anemia. The commonest anemia is which one? Microcytic, because of iron deficiency, because of diet, blood loss usually, either diet or blood loss, but mostly blood loss, because we lose a lot of blood through a lot of things. So you get small cells, the RBC becomes smaller, but here the RBC becomes big, so it's called a macrocytic anemia. And normochromic, normal color. Kalau microcytic itu biasanya hypochromic, a bit pale, right? And here is because of vitamin B12 deficiency. But why you get vitamin B12 deficiency? Because there are other causes. So there's some erroneous statements in the literature, right? Because people often get confused about pernicious anemia. So what is actually pernicious anemia? Pernicious anemia is an autoimmune disease. The immunity attacks the
red blood cell.
intrinsic factor, right? If you remember how your acid is being produced by the parietal cells, intrinsic factor, right? So when you have your immune system attacking intrinsic factor, you don't have intrinsic factor, you don't get vitamin B12 being absorbed. Even you makan vitamin B12 yang banyak pun, right? You don't have these factors. So this is pernicious anemia. It's different from B12 deficiency in diphyllobothriasis. This is because you have a big worm there that's absorbing competitively vitamin. Each time you makan, dia makan dulu. Your intrinsic factor is fine. It's just because there's a worm there that's absorbing all the vitamin B12. So don't confuse this. So both results in lack of B12 deficiency, so you have anemia. You don't have B12, so you have anemia, which is large cell, megaloblastic, macrocytic, right? So we always ask this in exam, right? You have to explain. So you have to know the difference between pernicious anemia, right, which is an autoimmune disease. So it results in failure of vitamin B12 absorption. Where diphyllobothriasis, simply the worm is absorbing all your, competitively absorbing all the B12, right? Everything else is intact. So that's why, for some reason, right? That's why it can survive for so long. It becomes so big. It has managed to evade the immunity, right? Although big, the body simply doesn't detect it, right? So that's why, technically speaking, it doesn't eat, apart from vitamin B12, it doesn't really have much problem other than just generalized gastrointestinal symptoms, right? Tapi kalau it becomes too big, right, more than 10 meters, what can happen? Something so huge in the intestine. Obstruction. So sometimes it becomes maybe you get intestinal obstruction and it needs surgical removal, right? Tapi vitamin B12 pula, if it's chronic, right? So you have symptoms, fever, glossitis. What is glossitis? Inflammation of the tongue, right? Paresthesia of the limbs. That's the birth and so on. So you see, some of the cells are bigger, right? It's not normal size. Glossitis. So how do you diagnose? You need to find this segment, because the segment is being detached, right? Or you may find the ova, right? So the characteristic segment is with the rosette uteri, right? With empty, usually with no ova. So treatment, you start to see Actually, you see, you can use the same treatment with nematodes, right? Praziquantel is an antihelmintic, right? Particularly effective for all flatworms, right? All flatworms except fasciola, which you have to use triclabendazole. But other than this, all flatworms, either trematodes or Cestode, responds well to praziquantel. Niclosamide can only be used for those in the lumen, right? Because very few gets absorbed in the circulation. So yang mana yang dalam intestine, you can use niclosamide. So like Diphyllobothrium and tinea and so on, yes, you can use niclosamide, but nowadays praziquantel is the drug of choice, right? Niclosamide is an older drug, right? Prevention, because it is being passed in the feces into the lakes, right, in the river. So you should not do that, or should not prevent from getting infected by cooking the fish, right, or freezing the fish. Deep freezing also kills the worm. Deep freezing, but not salting, right? Like I said earlier, salting, jeruk, peram, pekasam, sebenarnya doesn't kill the pleurosarcoid larva, but deep freezing does, right? Tapi the lower the temperature, dia lagi cepat, right? So for example here, if you minus 10 macam ni, right? Minus 10 ni dapat kat mana? You punya peti ais tu berapa temperature dia?
Negatif.
Ha?
Negatif.
Pasal tak? Ada range lah. Tapi when we say fridge temperature, it is 4 degrees. But of course you can play with the range. Lepas tu you ada freezer. Freezer can go up to how much? The normal freezer yang fridge tu, yang datang dengan fridge, not the another freezer. That one can go up to minus 10. Tapi kalau yang Alex satu yang buka yang macam orang simpan frozen food tu, can go even further, right? So ada yang sampai minus 20, but usually minus 20 ni for lab purposes. For lab specimen, we have at least for minus 20, and even we have for DNA studies semua minus 80, right? Tapi tu semua tak. Yang untuk makanan punya ni, at least kat rumah minus 10 or minus 20 tu dalam gudang tu. So you can see minus 10 for at least 2 days. So biasanya industri ni akan bekukan dulu, right, for many weeks. So usually selamat lah. But still you have to be careful, right? Or you cook more than 60 degrees. Kalau macam gitu-gitu je masak kan, sate dan sebagainya tu yang tak masak, right? It have to be more than 60 degrees. Kalau more than 60 degrees tu, more than 10 minutes tu dah okay, right? 10 to 15 minutes. Kalau lagi tinggi, lagi cepat, right?